Hello, everyone. Thank you for joining us today for this session. Welcome to our Ask the Expert series. If you're new to our webinar series, welcome and thanks for being here today. Criterion Edge runs multiple informative webinars throughout the year. Today, we will be doing a live 45-minute question and answer session on understanding clinical data sufficiency and strategies to address limited clinical and performance data. We're proud to announce that Criterion Edge has been awarded Company of the Year among top regulatory services companies. So how can we help you? If you'd like to speak with us about your upcoming projects or regulatory writing needs, we would be happy to book an appointment with you. You can email us for a free 30-minute consultation at consult@criterionedge.com. We have two presenters today. Welcome, Stacy Beecham, Principal Medical Writer with Criterion Edge. Stacy has a BS in Biology and an MS in Human Physiology with extensive experience in regulatory writing and 10 years of experience in international regulatory affairs. Stacy was a medical researcher prior to entering the regulatory space, where she worked with physicians and scientists at Mayo Clinic in Minnesota. Stacy's expertise in regulatory affairs, submission writing, clinical research, and regulatory consulting for medical device, biopharma, and biotech makes her a valuable asset to our team. Also joining us today is Dr. Sarah Chavez, Director of IVD and Scientific Writing Services with Criterion Edge. Sarah has a BS in biochemistry and a PhD in molecular and cellular biology with 25 years of laboratory bench experience and has taught at the university level for 20 years. She has extensive scientific writing experience in an academic environment, as well as for several large textbook publishers. Sarah's strong background in basic research and her extensive experience with regulatory writing will allow her to assist companies with IVDR readiness as part of the Criterion Edge team. And now I would like to welcome Sarah to give an overview of how our session will look today. Hi, Sarah.
Hey, hi. Thank you, Olivia. And thank you, Stacy, for joining us as well. So in today's session, we realized through our previous two webinars that we hosted that there are still a lot of remaining questions when it comes to well-established technologies and all of these different topics. So today we're hoping to answer some of the remaining questions regarding how to address common gaps in data sufficiency and how to identify them in the first place. Some of the strategies to effectively evaluate clinical and performance data, and how to approach the CER if you do fall under one of these categories of legacy, wet, or devices that are exempt from clinical trials. And hopefully we'll build in some of those best practices when you're actually approaching the task of writing the clinical evaluation report.
All right, let's get to our first question here. Let me know when you can see it on your screen.
Okay, yes.
All right, so our first question today comes from Severine, and she's asking, when do we know data are sufficient?
I can start with this one because this is actually one of the most common questions we get. And unfortunately, the answer is you don't really know until it goes through the process of being evaluated. But you can take some proactive steps to ensure that you are putting forth the best possible report and product. So as a rule of thumb, I just tell our clients to make sure that we have exhausted all sources of data, that we've very clearly documented it, that you are solid on your template and your strategy that you're going to take, and that you've also really thought out how to design proper searches and strategies to capture the most data. But then as you're writing the report, you also want to make sure that you're including that justification of why did we make the decisions we made very early in this process to seek out and proactively collect all of the data that's available. And if you do identify gaps, be willing to acknowledge those and address how you're going to fill those gaps in the future reports.
All right, thanks, Sarah. Let's get to our next question. This one comes from David. He's asking, how do the requirements for clinical investigations differ between Class 1 and Class 2A devices? So what would you have to say about this?
I can take this one. David, great question. This comes up a lot as well. For class 1 devices, sometimes you can just self-certify and there's not the requirement for a notified body to review your clinical data, but in many cases, your device may fall into this device subset of device groupings within class 1 that does require review by a notified body. Typical process for class 1 devices would be to analyze the published clinical data from literature. And that way you can determine your safety and performance measures that your device will need to meet to show compliance. For class 2A devices, on the other hand, it's quite different. They definitely require review by a notified body. And you are required to have device-specific clinical data to demonstrate compliance. If your device is a wet device or a legacy device, you could use older clinical data and combine that with post-market data to show compliance to the MDR. But if it's a newer device, you'll probably have to conduct clinical trials most of the time for class 2A devices.
All right, thanks, Stacy. Moving on to our next question. Stephanie is asking, is clinical data required from published clinical trials, or is non-published data also sufficient?
So I think we can kind of tackle this by breaking down the question and sort of thinking about what you're really asking. Non-published data, so internally held data that is manufacturer held, is definitely something that you're going to want to present, and it's very commonly presented. As far as sufficiency, though, you wouldn't necessarily want to only present manufacturer-held data in lieu of published clinical trial data. In order to ensure that completeness and that sort of unbiased nature of the systematic literature review process, it's necessary to also show that you have sought out any published data or clinical data regarding your device. So like, for instance, let's say your in-house data presents your device in a very positive light, but there is clinical trial data that seems a little bit less positive, and you can't just justify excluding that data just because it doesn't present you positively. That's not systematic and that's not unbiased. So I would say sufficiency requires that you seek out all sources of data without picking and choosing which data you're going to present.
All right. Our next question, Sarah, is about state of the art. This question is from Alice. How much and what kind of evidence is sufficient for the state of the art?
So this can definitely vary from report to report. I would say, generally speaking, the state of the art should describe what is the current view within the field based on experts in the field. So I typically start with a review of clinical practice guidelines and recommendations and things like that to get a high-level overview of what experts in the field view. And then I build from there. So it's going to require that you fully address all aspects of the PICO terms that you've laid out. So your patient populations, your indications, your comparators, and your outcomes. When you're, so I would say that would be the biggest thing to be on the lookout for in terms of sufficiency within the SOTA. Make sure that if you have an indication, you address it within the SOTA. If there's a specific population you're covering, that should also be presented and described within the SOTA. So the state of the art should be broad enough to capture all of the data that's out there, but also specific and tailored to include all aspects of your device's PICO terms.
All right. Moving on to our next question from Tim. What are the specific differences between wet and legacy devices? Would it make sense to argue that a device is both?
Sure, I can take this question. So legacy devices are any devices that were previously certified under the MDD or the AIMDD. And then wet devices have four specific criteria that we discussed in the previous webinar that all wet devices have to meet to be considered well-established technology. So yes, definitely there are many devices that could be considered both a wet and a legacy device. Just please look back. I think that Olivia will probably put this in the chat, the link to our previous PowerPoint presentation, so that you can review those criteria for the wet devices.
Yeah, I'll pop that in the chat right now.
Thanks.
All right, moving on to our next question. What are the expectations for devices with no direct clinical benefit, like, for example, a catheter?
Sure, I'll take this one too. I imagine you're referring to a system, right? Where a catheter is part of a therapeutic system. And so it has an indirect clinical benefit in that it works together with other devices to provide the benefit to the therapeutic mode to the patient. For example, if you were talking about like an introducer that introduced a catheter that then reaches the blood vessel and inflates the balloon and treats the blood vessel, the introducer's part used as part of that system, but it doesn't have the direct clinical benefit. It facilitates the mode of action that provides the direct clinical benefit. But if you're talking about a catheter that, for example, is used for drainage or urinary purposes, it does, in effect, when working together as a system, it does provide the direct clinical benefit. So you would talk about, in your clinical evaluation, you would talk about data that refers to the whole therapeutic system and then outline the direct benefits and indirect benefits of each component or each aspect of that system.
All right, thanks, Stacy. Our next question is from Virginia, and she is asking, what is the strategy to predefine sufficient evidence of clinical data in the CEP?
So I think for this one, again, it kind of to take a step back, as you're stating, the purpose of the CEP is to show your plan. How are you going to seek out proactively additional data as it is newly published moving forward? So you can't really predefine what is considered to be sufficient within the CEP. You don't know what you're going to find until you start looking for it, really. What you can do is make sure that you're including a detailed description of the methodology of your search strings. You're including all the different databases that you're going to be seeking out, certain organizations that you're going to be searching through. So you want to ensure that you are casting a wide net to capture all data that may arise, but you also don't want to be too limiting, just given that the technology could change, and you want to make sure that you're capturing that in your updates to your search. So for the plan, the key is to make sure that you're being detailed in the methodology that you are proposing to capture sufficient evidence.
All right, thanks, Sarah. Our next question is from Simon. And, oops. He is asking, our device has very limited clinical data for one patient group. Is add-in PMCF survey enough? So what would you have to say about this situation?
Yes. Stacy, did you want to cover?
You go ahead, Sarah.
Oh, sure. Okay.
So
the PMCF is certainly a good approach. It never hurts to add in a PMCF survey to ensure the completeness and the sufficiency of that data set. This is a fairly common issue, especially if your device has been on the market for a while, or if it's being used for a purpose that wasn't originally intended. But if it's part of your intended purpose and it's part of your IFU, you are expected to have some clinical data with regards to a particular patient group. So the PMCF survey will help to address a perception of data insufficiency, but definitely build that into your searches. And in the event that you're still not capturing that data, it may ultimately be necessary to design a targeted study to directly address that.
All right, thanks, Sarah. Here is our next question from Irina. How do you identify a listing of similar devices to be included in the clinical documentation?
Sure. Similar devices, identifying similar or competitor devices, can be used in a variety of ways. But if you're trying to identify similar devices for establishing the therapeutic landscape or therapeutic alternatives, or for your state-of-the-art aspect of the CER, one good way to identify those would be to look at classification, device classification. So if your device is a class 2A device and falls under Rule 9 or something like that, you want to look for other devices that would have a similar classification. Sometimes this information is easier to find for certain devices, and then for others, it's more difficult to find. If you cannot find classification information, I would recommend that you look into the intended use, which you can find in the instructions for use, the IFUs for the various products, and then the intended population. So those are two main aspects that would identify similar devices. So if you can find a handful of devices that you think might be similar, then dig into their IFUs on the websites and look at that intended use statement. Make sure that it's aligned with yours or similar enough to yours, and then that the intended population, of course, adults, pediatrics, neonatal, what have you, is similar, then you may be able to use those device criteria to set your acceptance criteria for your safety and performance measures that your device will be compared against to show compliance to the GSPRs.
And I can jump into just from the writer's perspective. I know that as we're scoping different projects, that similar term really gets us into a lot of issues, because similar can be defined in many different ways. It could be like a predicate device. It could be something you're claiming for equivalence. It could be an alternative, or it could be a competitor. So I'm always very purposeful in avoiding the word similar when I'm trying to plan out a CER or an SOA section with our clients, to make sure that I'm being very clear on where that data is going to be included and how it's going to be used. You wouldn't want to include, if you're defining similar as an alternative or a competitor, that would not be part of your own device's clinical documentation. You can't use a competitor's device, the data from a competitor's device, to justify your own device. You would include that in the competitive landscape within the state-of-the-art section. And that's going to help you derive your acceptance criteria by which your device will be judged. So it's really important. And the other thing to point out is that a lot of times clients will give us a really long list of competitor devices to include. I would definitely recommend narrowing that down to maybe the top 3 to 5 devices. I've had some companies that have wanted to use upwards of 20. That can get very time consuming, and it doesn't ultimately affect your acceptance criteria in the end, but it can result in a really long process. So I would say being purposeful in who you're selecting as your competitors to be able to derive your acceptance criteria. Go to the literature, go to clinical practice guidelines, look at some of the studies out there that are using sort of a benchmark to compare new technologies as they're coming out. See who seems to be the most widely published or the most well-respected within the field. And those are good places to start. Sometimes we'll get some tips from the marketing groups, but you want to identify the devices that are absolutely the closest to yours so that your acceptance criteria are allowing you to measure your device based on something that's very, very similar to your device.
All right, great perspective, Sarah, and great advice, Stacy. We're about halfway through our session, so I want to remind everyone to keep on sending us your questions using that Q&A button on your Zoom menu bar, and we'll keep on going. This next question is an interesting one. What can we do for a device that has data for one use environment but not another? We are submitting for MDR.
Oh, okay. So I think this was one that I saw in our Q&A button. This I've encountered quite a bit when, like, let's say you have a device that's intended for use in the central, like in the carotid, and now you're trying to also claim it can be used in the periphery, or peripheral vascular system. And then you start digging into it and you realize I'm not finding any data on this other intended use. That can be tricky. You will definitely want to address that, and that may again require you to set out some descriptions in your plan about how you're going to proactively collect that data. If you know it's being used in the periphery, you may look to some registries or some other sources of published data, or you may set out to do a short study to help supplement the data that may not exist in published literature. But yes, definitely make sure that as you're collecting the data and using that for the acceptance criteria, that you're being purposeful in collecting data for all different environments, not just a single environment, even though one may be much more highly represented in the literature.
All right, thanks, Sarah. Here's our next question from Georgiana. Is a clinical development plan applicable for a legacy device?
Sure, I can take this one. The short answer is a clinical development plan is required for all devices, but legacy devices are specifically outlined, and good examples are given in this MDCG 2020-6 document that I just popped the link to in the chat. So I think the clinical development plan, in its simplest terms, outlines the essential requirements that you need to meet, and how you're going to go about analyzing the clinical data, collecting the clinical data, evaluating the clinical data, make sure that you are conforming to the GSPRs. And then, of course, you need to outline your intended purpose, target groups, indications, contraindications, warnings, describe the intended clinical benefits, and then specify your qualitative and quantitative aspects of your clinical safety and performance outcomes. So those are the main aspects of a clinical development plan. It's saying, hey, how are we going to go about evaluating the clinical data and the clinical evidence and information, associated with this device? For a legacy device, it's basically taking the original clinical development plan or clinical evaluation you used, and then updating it to be sure that it meets the more stringent requirements of the MDR. So you'll be taking what you used for the previous certification under the MDD and AIMDD, and then adjusting that clinical development plan to make sure that it's now in conformance with the new requirements, and then showing how your post-market data will, maybe if additional clinical evidence is required or data collection is required, you'll show how you're adjusting for that for the new requirements.
All right, thanks, Stacy. Here's our next question, and we get questions on software as a medical device pretty often. So is the software developer of a SAMD or a SIMD responsible for preparing the adequacy of clinical data and clinical evaluation report? So Sarah, what would you have to say to this question?
So in my understanding, the software developer would not be required to present the clinical data as part of the CER. They should provide the validation data of the software, but that's going to be more of the sort of like the analytical data. It's not going to be the real world clinical data. The manufacturer who's preparing the clinical evaluation report would be required to seek out the clinical data pertaining to the software.
All right, got it. Here's our next question from Thelma. And she's asking what to do in the absence of clinical data for a new environment of use.
Yeah, I think that seems to be a really common question. When you don't have clinical data for a particular environment, whether it's a particular anatomy site or a particular patient population, or even a particular clinical scenario, it's going to be really important to set up a way to address that in your clinical evaluation plan. So think about how you're going to collect that data moving forward. Is it just that the field itself has not used your device for that purpose up until now, and this is a new indication that you're going to be rolling out? Or has it been used up until now for off-label or like sort of the off-label use of your device, and you may have data out there that you're unaware of. So it really depends on the situation, but I think having a solid plan to continue to collect that data through both systematic literature review as well as your PMCF surveys, or that part of that follow-up, that's going to be really critical to help you start to build that clinical data source that you're going to be looking for.
All right, thanks, Sarah. Moving on to our next question. What is the role of residual risks like a new AE towards clinical safety and performance?
Oh, this is something else that we often encounter too. When we are tasked with writing, whether it's an IVDR where we're writing the scientific validity report, or if it's in a CER where we're writing the state-of-the-art section, we're on the lookout for meaningful clinical safety and performance objectives. We want to make sure that we are capturing data that's going to inform us of the risks and benefit profile, but we also want to make sure that that data helps us support the risk benefit analysis for your particular device. In the screening process, you may actually start to identify new potential adverse events, or maybe there are specific contraindications or a particular event that's newly identified. By looking for case reports or case studies, while you won't include those necessarily as part of your landscape analysis, those can be really insightful as to a new potential risk to be on the lookout for moving forward. So we tend to include case reports as an exclusion criteria when we're doing our searches, but we don't eliminate case reports from our search strings, because those are going to be a really solid way to identify the potential for risk, and then to make sure that the discussions you include in your state-of-the-art section align with any residual risk or the risk benefit analysis that you're going to discuss elsewhere in the CER. So it's all about making sure that you are justifying any residual risk as it pertains to the benefits of your particular device based on what the competitive landscape is telling you.
Great, thanks, Sarah. Here's our next question. Could you please just briefly explain what benchmark device means?
Sarah, do you want me to take this one, or?
Oh, sure.
Have it. Okay. Well, Sarah, actually, you'd probably be able to explain it better in the framework of CER for CER purposes, maybe.
Yeah. I think this is kind of funny because we all use the term a little bit differently.
Yes.
When I am writing it as a writer, I think of a benchmark device as something that is considered to be sort of the standard, like the gold standard within a field. So something by which other devices are commonly measured. So I tend to be on the lookout for these so-called benchmark devices to help me me identify comparators and also some of those important alternatives to be on the lookout for. So, some types of devices have like the standard that's like the standard of care. And, there may be additional newer technologies that are coming in, but the efficacy is usually judged against something. And that's what I would consider or classify as a benchmark.
Exactly. And sometimes you have, for example, like the NCCN guidelines for cancer devices, they'll outline the gold standard for brain tumor surgery or whatever. And so you can kind of get an idea of what the benchmark would be in each therapeutic area by looking at the standards and guidelines that reference that area.
All right. Here's our next question from Brian. Brian would like to know, with common wet devices where literature is minimal, how do you justify insufficient data?
That's a tricky one. Do you want me to take it, Sarah, or?
Oh, sure. I would just say
Go
you
ahead.
can't really justify insufficient data. If the data is insufficient, that's going to be an issue. I think it's all about the approach that you take if you are determined that your device can fall under well-established technology. From a writer's perspective, I would go back farther in the literature as I'm doing my state-of-the-art search. It could be that if this is a well-established technology and I'm only looking for the past 5 years, I'm not going to see a lot of new data published in the past 5 years if this device has a market history going back 20, 30, 40 years. So instead, I'll be on the lookout for maybe some systematic reviews or meta-analyses that help me justify the history of the device and how it's performed over a long, extended period of time. So it's about sort of like strategizing the approach you're going to take to looking for the literature by being able to anticipate how commonly published that data is going to be in different time frames.
Exactly. And adjusting your post-market clinical plan to collect more data, that would be, yeah.
All right, thanks for that advice. Moving on to our next question from Ash. How can we devise general criterions for evaluating data sufficiency while acknowledging variances between devices?
This can be really challenging because you may have some companies where they have one device that has way more data than they know what to do with, and then other devices where there's virtually nothing. So it's sort of this feast or famine problem. We've had instances where we want to maintain the data sufficiency by casting a broader net and making sure that we are going back farther in our searches, or including data that may have smaller sample sizes. Like if it's a rare disease or something, you may be using studies that only include 5 or 10 people in some of those case series. Whereas if it's a very, very commonly used device, you would want to limit these studies to maybe more than 100 people or even larger than that in some instances to help you ensure the highest level of evidence. So it's about finding that balance. You want to make sure that you are able to justify the choice of data that you're collecting, to clearly present the appraisals of the data so that you're showing that the evidence that you are collecting is of the highest quality available. And then also being really clear in the methodology that you're going to take to fill in any existing data gaps or sources of that maybe, I wouldn't call it an insufficiency, but an area where you know that there is limited data. How can you be proactive in collecting additional data or designing studies to fill in some of those gaps?
All right, thanks, Sarah. Here is our next question from Karen. Does a Class 2B standalone software without direct safety concern to the patient require clinical data? Stacy, you want to take this one?
Sure. Class 2B standalone software, I think that the best approach here would be to consult the guidance because there are so many new stipulations around software and standalone software that's regulated as a medical device as part of a system or not as part of a system. And I think that once you go through this MDCG guidance that I just put in the chat, making sure I did the right one, it should be MDCG 2020-1, I believe. So let me make sure that yeah, that's the right one. Look through there, and it helps to kind of help you classify it, and then it tells you what kind of clinical data would be required if it really is standalone, or if it's used as part of a system, or if in some cases it could be sold separately, but also used as part of a system. It depends on if it's diagnostic software, or if it's related to imaging, if it's related to therapy, if it has indirect clinical benefits. All of those things are factors that you'll need to consider. So it's a great question, but I don't have a direct answer. And so you'll have to just comb through the guidance and kind of see where it falls.
All right, thanks, Stacy. Here is our next question from Simon. Can clinical evidence only be sufficient for a low classification device like a class 2A if clinical data is not available in the clinical literature?
So I think what this is asking is basically some of that indirect evidence. You know, if you have a lower classification of device and you don't anticipate that there's going to be a lot of clinical data out there, then you can use some of these sort of indirect ways of measuring it. So whether it's part of a system, or if there may be other ways of, it could even be like technical success, like, you know, how often does the device perform as intended? So there may be ways to look for the device being used in clinical literature, but you are definitely going to be more limited if it's just not something that you would anticipate being published on very often. So there, I would definitely have you rely on the clinical practice guidelines or recommendations by organizations, but also make sure that you can find some of that sort of indirect clinical evidence.
Okay, thanks, Sarah. We have a couple minutes left, so if you haven't sent in your question, please do it soon. We'll try to get to a couple more. Here's the next one. Can you please talk about the impact if there is no clinical data on the technical file? Stacy, do you want to take this one?
Sure. I guess it depends on whose technical file you're referring to. If you're referring to an equivalent device that you're trying to measure up to, and you can't find any clinical data in that technical file, or if you're referring to a previous generation of your own device, there again, I guess I would look for published clinical data from trials that used your device since the time that the submission that used the technical file was established. So post-market data, real world evidence, those types of clinical data would need to be supplied in order to demonstrate compliance under the MDR.
Got it. All right. Moving on to our next question. How do you set acceptance criteria if you do not have a very similar device on the market to use as a comparator?
This can be really challenging. And it really depends on sort of how your device is positioned within the therapeutic landscape. So I would suggest ask yourself what have been used before my device was here. Like, what were people using as that benchmark, as that gold standard for the treatment? It could be that there really wasn't a whole lot of treatment options out there. And in that case, you would have to state basically how well does this type of device, does this type of treatment perform compared to doing nothing. It could be that, like, for instance, let's say there were medications that were previously used, and now you have a device that can more directly address a clinical condition. In that case, you may be comparing yourself to some of these state-of-the-art alternatives rather than a direct comparator. So that can be one approach that we've taken if the technology is new, or if you are sort of a standalone device in a type of technology where there really isn't another direct comparator. That's where you're going to rely heavily on your state-of-the-art literature.
All right. Thanks, Sarah. Here is our next question from Anna. We have clinical studies completed as per IVDD Annex 1, as well as data from outside of the EU. Can we use those data?
Yes. All sources of clinical studies are definitely helpful. For many of the IVD clients, they're dealing with a complete lack of clinical data. So I would say yes, but you want to make sure that you are careful to point out that these data are obtained from outside of the EU. If the incidence of that testing is similar in if it's something like pregnancy testing, that's going to be very similar across different countries. If it's something like HIV testing, where the data collected may be very different, there could be different types of viruses or something like that in one country versus another, or COVID testing, like different strains of the virus that are circulating in one versus the other. I would be careful to directly address the epidemiology of the disease and show that the data from another country is similar or can be extrapolated to also cover the EU population. So just be very careful about justifying that the population level data can be supported.
Okay, great. I think we have time for one last question. What do you do when the IFU is from 2015 or 2016 and you cannot find more actual data? Maybe the intended use or intended population has changed. Where do you find more actual data and where?
We have actually encountered with some
Yeah.
clients. Yeah, this is funny. Like, we've had some clients where they're looking for IFUs and they literally have scans of PDFs from 20 years ago. So 2015 to 16, you're actually doing pretty well compared to some that we've encountered. If there is searching out additional actual data, I would say that this is where it's really important that you make sure that your IFU is still current, that it doesn't need to be addressed prior to writing the CER and updated. If the intended use and population have changed, you need to first change your IFU, because everything that you're going to write in the CER is predicated on clearly defining your intended purpose. So if that's not clear, or if that's going to undergo revisions, that needs to happen very early in this process so that you can then design your searches and your strategy and your overall approach to be aligned with the current IFU.
All right. We are out of time. I hope that Sarah and Stacy were able to answer most of your questions, but if we didn't get to yours, we will follow up with you by email later on. If you'd like to discuss your question or your project needs further, we invite you to book a free consultation with us. Send us an email to consult@criterionedge.com to book an appointment. Thank you again, Stacy and Sarah, and everyone for joining us. There were so many great questions answered here today. Take care, and I hope to see you at our next webinar.